It actually has an internal hydrostatic skeleton. So if we take a look at here, the spinal cord, it's not shown, but now it is. Right in the middle of the spinal cord, there's a hole. That's the central canal. And that connects right here to your fourth ventricle. So the fluid-filled cavities of the brain and that central canal of the spinal cord, you have cerebrospinal fluid circulating through it. And these are the cells. These are the ependymal cells that circulate that stuff around. And the cilia would be here. They're just not shown. So they have cilia facing the lumen of these spaces circulating that stuff around. So that's the ependymal cells. Next, we have astrocytes. They're the most abundant of all the glial cells, which means they outnumber neurons. Neurons, by the way, are outnumbered by glial cells 10 to 1. So there's 10 times more glial cells than there are neurons. So astrocytes are actually the most abundant neuron in your central nervous system. And they don't have neural function really at all. So what they do is they help form the blood-brain barrier. They have these vascular feet called pedicels that help them do that. They lay down some connective tissue when neurons die, like following a stroke. So they do some wound repair. And then there's scavengers of neurotransmitters and ions that are in the interstitial space between neurons. You'd have to imagine there's millions and millions and millions of neurons in your brain. There's a lot of neurotransmitters floating around in the ISF and ions. So what helps pick these neurotransmitters up and ions and re-deliver them back to neurons are astrocytes. Let's take a look at them. So here is an astrocyte right here. You can see it's got these long projections here. And at the end of its cytoplasmic projections, it's got these little feet. These little feet are called pedicels. Now, these projections here, we're not going to go into, but look at these ones here. These pedicels are forming around a blood vessel, a capillary in the brain, and it almost completely covers it. And what I mean by it, several of these pedicels from astrocytes just butt up against the blood vessel and kind of surround it. And so if you looked at a blood vessel laterally like this, there's your endothelial cells that come from what germ layer?
The endoderm?
Good guess.
Oh,
Mesoderm?
Correct. So they
oh
come
my
from
God, right,
mesoderm.
right, right.
So look, things can get out of capillaries pretty easy by going between endothelial cells, but in the central nervous system, we have this blood-brain barrier that makes it difficult for things to get out of blood vessels. We don't just want any chemicals getting out of our bloodstream into our brain. And the reason for that is not only do you have to get between these cells, you have to get between these pedicels as well. So this is the blood-brain barrier right here, people. And so we design drugs to try to get around this. And it's a very difficult thing to do. And so difficult that most chemotherapeutic drugs, if you have a brain tumor, we don't even bother with doing anything other than cutting a hole in your cranium and just putting a port in your brain and just delivering the chemotherapeutic drugs that way. So that's how tenacious the blood-brain barrier is. So you can also see the pedicel forming connections or the astrocyte forming connections via pedicels with neurons as well. And this is that scavenging of neurotransmitters and ion functions that they do. The third one is the oligodendrocyte, probably the one you're most familiar with. This myelinates neurons in the central nervous system. So it's forming a myelin sheath, which is basically a lot of plasma membrane. It's wrapping its plasma membrane around axons of neurons. And one oligodendrocyte can wrap several different axons, really several different regions of several different axons. And where it begins wrapping and then it begins again, there has to be a space where there's no myelin. That's called a node of RANVA. So let's take a look at that. So here is an oligodendrocyte. Here it is extending its plasma membrane, and you can see it's wrapped it around this portion of the axon here. And another one is wrapped it around this portion of this axon here. So there's a portion where there is no myelin. That's a node of RANVA. So where it's myelinated, we call that an internode. And so it's a bunch of plasma membrane. Now, it doesn't look like it. It looks like maybe the oligodendrocyte forms a hot dog bun, and then the axon just sits in there like a hot dog. But it's not really that. Take a look at all these little lines here. All those little lines are layers of plasma membrane. So it's like taking a piece of gum and wrapping it around your fingers several times. You're basically myelinating your finger with gum. And that's what that cell does. It wraps its membrane several times, about 7 to 10 times around an axon to myelinate it. And make no mistake, people, that's the insulation of our wires. So when you think about having insulation on your cell phone charger or whatever it may be, that plastic or rubber, in the human body, we don't have plastic or rubber, but we have something analogous to it. We have bilayer. It's hydrophobic, and so that's what's going on here, insulation of wires. Now you can see one oligo can myelinate a couple segments of a couple different axons. It can't myelinate an entire axon. Some axons are like a meter long, but it can do several regions of several different axons. And then lastly, we have the white blood cells of the central nervous system. We call them microglial cells. They are macrophages, so they do phagocytosis, and those are shown right here. Any questions about this? So that's the four glial cells of the central nervous system. There are two in the peripheral nervous system. One is called a satellite cell, and it surrounds neuron cell bodies in regions that we call ganglia. A ganglion is an aggregation of nerve cell bodies in the peripheral nervous system. Let me say that again. A ganglion is an aggregation of nerve cell bodies in the peripheral nervous system. So if you take a look here, if you blew up this region of the nervous system, this is called a dorsal root ganglion. We'll talk about it in a little bit. If you blew that up, what you would find is a lot of cell bodies of neurons there. So that is called a ganglion. Now, if you were to blow that up even more and look at a cell in a ganglion, just one of those neuron cell bodies, look at those cells hanging out and surrounding the cell body. We call those satellite cells, and we call them satellite cells because it looks like they're orbiting around these cells. But what they function like is astrocytes. Now, there's no blood-brain barrier or anything because we're not even talking about the brain. We're in the peripheral nervous system, but they do that similar function of scavenging neurotransmitters, ions, redelivering them to these peripheral neurons. And then lastly, we have the cells that myelinate neurons in the peripheral nervous system. So remember, oligos do it in the central nervous system. Schwann cells do it in the peripheral nervous system. And so they form these myelinated coverings. And then one Schwann cell can only myelinate one segment of one axon. So one Schwann cell cannot myelinate several different regions of several different axons like an oligo can do. So that's one Schwann cell there, another one, another one, another one. And what are those regions where there's no Schwann?
Aren't they nodes?
The nodes of Ranvier, right? And where there is Schwann, it's a node of sorry, an
Internode.
internode. And remember, it's not like a hot dog bun here. That Schwann cell is making several layers of plasma membrane and wrapping it around. Take a look at it. Is this an SCM or a TEM?
TEM?
You got it. Very good. It's a transmission electron micrograph. And what you're looking at, people, is here's the membrane of the neuron. That membrane of the neuron is called an axolemma. Just the special name for plasma membrane of a neuron, more specifically of the axon region. So that's axolemma, and this is axoplasm, just the cytoplasm of an axon. Now what you can see is the nucleus of a Schwann cell, and every single one of these black lines here is a layer of its plasma membrane. So that's how it works. Now, what's flowing in this axoplasm is not electrons, like in the charging cable to your cell phone. What's flowing is ions. but it's still electrical current, and we have to insulate that. And that's what we're doing here with 7 to 10 layers of plasma membrane. Is that a cool image or what? I mean, it just doesn't really get much cooler than that. I mean,
Wait, you
I mean,
said the nerve cells are in the exoplasm or the ions?
say that again.
What's in the exoplasm? The nerve cells or like the ions?
Well, no, this is a nerve cell. It
Yeah.
has an axoplasm. So the axoplasm is its cytoplasm.
So it's all just continuous. It's just really, really long and a spiral.
Correct.
That's insane to think about.
It really is. Really is. Now, we'll talk more about this later, but you already know about it, right? There's some conditions where demyelination happens, where this myelin can be wiped out, and now people have short circuits in their wires, like multiple sclerosis. So neurons, we can look at them structurally and we can look at them functionally. So let's look at the three general structural classes. There's a million different types of structures of neurons. If you want to Google one right now and be blown away, look at a Purkinje cell. So take a look at a Purkinje cell and look at its dendritic tree. It's insane. So there's many more than this, but these are the three general categories. So we have bipolar neurons, and structurally what that means is that you have a cell body and it's got two projections coming off of it. Now, it's actually got this one projection that's basically it's one of these dendrites. So these dendrites that are receiving sensory information, they collectively form a single dendrite, and then an axon comes off the other end of the cell body and forms these synaptic terminals. And we'll talk more about that in a minute. And then we have these unipolar neurons where the cell body only has one projection coming off of it. And then that projection splits. There's this peripheral axon where dendrites merge to, and then there's the central axon that forms these terminal knobs here. So the unipolar
Professor,
neuron,
what do you mean by projection?
these extensions you see here.
Okay.
Well, projection would be like if you look at the cell body here, it's got this membrane projection. That's what I mean by that. Does that make sense?
Yes, thank
Indeed. Now, if you take a look at the multipolar neuron, it's got, and I'll say that word again, it's got many projections coming off the cell body, and these projections are called dendrites. And that's why we call it multipolar, because it's got many processes or projections coming off of it. And then it's got this one, that's the axon, that goes out and forms these terminal knobs. So maybe you've noticed that this polarity has nothing to do with charge. So we were talking about plasma membranes having a charge being more negative on the inside than the outside. This has nothing to do with that. What this polarity means is how many processes come off the cell body. 1, 2, bipolar neuron. Got to look really closely. Just one here before it splits. Unipolar. Many, multipolar.
So we're usually multipolar when we're messaging, like when we're sending signals.
Well, that depends on where you're talking at. So
So
that
maybe-
I think your question will be cleared up on the next slide.
Okay.
So we can look at these neurons then functionally. So we can say that there are sensory neurons. So what they do is they receive information. For example, like if these cells had mechanoreceptors, or this cell had mechanoreceptors here, then you can have action potentials that form along this neuron and propagate to another neuron. So this is a sensory neuron. We also call it an afferent neuron. Now, where it's relaying its information to is what we call an interneuron. So this is 99% of all neurons in the human body are interneurons, kind of like a relay neuron. So now it's communicating through synapses with this neuron. This neuron will fire some action potentials, and then it'll communicate with this neuron, which is the motor neuron or efferent neuron. And when it communicates with it, it's going to fire action potentials. and then it's going to communicate with whatever effector it communicates with. So to answer the question previously, it depends on the type of signaling you're talking about or communication you're talking about. In other words, if it's sensory information, then it's going to be these neurons here. By the way, both unipolar and bipolar are sensory neurons. So please note that unipolar and bipolar are sensory neurons. But once you have sensed information, now that you want to make a command, like you want to make muscle move or something, then that would be multipolar neurons doing that. So these multipolar neurons are sending out signals to effectors for like muscle to move or glands to secrete, something like that.
So multipolar would be motor.
Yes. So this is a typical multipolar neuron. It's basically what we're going to focus on in this class. We will talk about sensory neurons as well, but the major focus will be the multipolar neuron. So you should know all the bells and whistles of that. I think I created a slide one day. It probably took me like 5 hours to create. So I just wanted all the components to be here. So you have the cell body of the neuron, and it's got a nucleus, and it's got Golgi and ER, all that junk that you would think a normal cell should have. But what it also has are these huge cytoplasmic projections. And these cytoplasmic projections, again, they're not cilia. They don't beat and move around and stuff like that. They're not microvilli. They don't absorb things. But what they do is the same thing cilia and microvilli do. They massively increase the surface area of the cell. So imagine what the surface area of the neuron would be if it didn't have dendrites. This would be the surface area. And now the surface area of the neuron is this. Right? It's enormous. So, and what do we want that enormous surface area for? For these little elevations on the dendrites that are called dendritic spines. Well, what connects with the dendritic spines? Well, nothing really touches them, but what interacts with them are synaptic terminals of other neurons. So synaptic terminals of other neurons are going to synapse on dendritic spines. So when you look at all the dendritic spines on this dendritic tree, you can start to get an idea of how many different neurons are synapsing on this neuron. And the answer is like thousands. So there's like thousands of synapses occurring on this neuron from other neurons. And those other neurons are telling this neuron do something, and other neurons are telling it don't do something. It's a combination of those signals that are coming to this neuron. By the way, in the form of neurotransmitters. And we'll get to that more in a little bit. Now, let me just show you something really quick, and then we'll come back to the details. This neuron then forms an axon, and that axon eventually branches into these terminal knobs. Now, what I want to show you is, here's another neuron over here with a cell body and dendrites, and here's its axon, and here's its terminal knobs here, here, here. And then here's another neuron over here, cell body. Here's the axon, and here's its terminal knobs. So in this context, then, we call this neuron the postsynaptic neuron. And we call these neurons presynaptic. So that means that this neuron comes after the formation of these synapses. So these are presynaptic neurons. They come before the synapse. This cell becomes after it. And when this cell communicates with other neurons, let's say another neuron here it communicates with, this is now the postsynaptic neuron. So I've drawn a couple of presynaptic neurons synapsing on this neuron, and look where they're synapsing. This one's synapsing on dendritic spines on the dendritic tree. This one's synapsing there as well, but also right on the cell body. So synapses can occur on dendrites and the cell body.
So they can be pre and post synaptic?
Well, again, that depends on what, well, you need some context there. If you have, for example, a neuron here, and then a neuron here, What's neuron 1 and what's neuron 2?
Is one postsynaptic?
One's presynaptic,
Oh,
because this is the synapse. This is the neuron before the synapse. This is postsynaptic, the one after. Make sense?
yeah.
Okay. So this is a very important region that we'll dissect out more later. The last portion of the cell body is called the axon hillock. It's a little cone-shaped region. So it's kind of a misnomer. It's called an axon hillock, but it's actually the last portion of the cell body. And then this one's named very aptly initial segment region of axon. So that makes a lot of sense. It's the initial segment region of the axon. So this is a very important region that we'll talk about later. This is where action potentials are going to be initiated. How many people have heard of action potentials before?
Yeah, I have.
Yeah.
Okay. Now, once an action potential is initiated here, it'll form here, then another one will form here, then another one here, another one here, another one here, here, here. And so where they form is at nodes of RANVA. Where they don't occur is at internodes. And we'll have to talk about what's occurring at internodes a little bit later. And you can see that there's a bunch of Schwann located at the internodes. That's why action potentials can't occur there. You can't have any current moving through a cell that has 7 layers of bilayer on the top of its membrane. So no currents moving here. Minimal currents moving, but you're not getting an action potential. Sometimes the axon will branch. We call that an axon collateral. We'll talk about why we might have one of those later. And at the end here, this axon is going to branch into what we call telodendria. And then these telodendria are going to terminate in these knob-like structures that are called knobs, synaptic knobs or synaptic terminals, either way you like that. Now, what happened here, while this was all going on, what we're looking at is the neuron was making neurotransmitters here, and it was transporting them all the way along this axon, storing them here. And now what's going to happen is those neurotransmitters are going to be released onto the dendrites or cell body of what kind of neuron?
Postsynaptic neuron?
Yep, a postsynaptic neuron. And is that a multipolar, unipolar, or bipolar?
Multipolar?
Indeed. So if you were walking along this neuron, and I'll do that to you on the exam, I'll make you microscopic and put you inside the human body somewhere. If you were walking along this internode and you fell and you ended up standing here, what would you be standing on?
A node?
Yeah, so you this is the node of RANVA, but what physically are you standing on?
The axon?
You are standing on the axon, and so it's just be a little bit more specific. What part of the axon?
The myelination.
Not myelination,
Myelination.
Fiber.
because you were standing on myelination here.
Schwann cells? Oh no.
Yeah, you were standing on them here.
Is it the nerve fiber?
No, you're standing on the axon. Someone said that's correct, but just literally, physically, what is your feet touching?
The axon collateral?
Not the axon collateral.
The not myelinated part of the axon.
Yeah, which is axolemma, right? Just the plasma membrane of the axon. So you're standing on the plasma membrane of the axon. Now we call axons nerve fibers. Now that doesn't mean we call entire neurons nerve fibers. We don't. Just their axon. And again, we call them nerve fibers because they're really long and thin and they look like a protein fiber. Just like why we call skeletal muscle cells protein fibers. Or muscle fibers because they look like protein fibers. Now, inside of the axon is axoplasm, and that's where we have a bunch of ions and, cell machinery that like we talk about in introbio, like microtubules, cilia, stuff like that. Okay, so let's talk about a synapse. So this is a neuron-neuron synapse, very typical. So the N1 cell is a neuron, and it has a synaptic terminal. And the N2 cell is either the cell body or dendrite of that neuron. It looks like this. So once we form these telodendria, they terminate in terminal knobs. So here's a synaptic terminal, otherwise known as a terminal knob. So that's the presynaptic membrane there. We don't consider so this is the presynaptic cell, but we don't consider the presynaptic membrane to be the entire plasma membrane of the neuron. Just this one that's involved in the synapse. So that presynaptic membrane is pretty much the membrane of the terminal knob. And what we find here are synaptic vesicles. These are membrane-bound vesicles that contain neurotransmitters. So the cell body manufactured neurotransmitters and it put them in vesicles, and the vesicles traveled all the way down the axon, and they were hanging out, that is, in the terminal knobs.
Wait,
So
so
something
the,
has to stimulate them to be released. Go ahead, question.
sorry, so the electrical signal, the action potential is what stimulates them, right?
Correct. And
Yes.
we'll go into a little bit more detail on how that works, but in general, yes. So let's just say that we have an action potential, and then what happens is these synaptic vesicles will fuse with the membrane here and undergo exocytosis, and now neurotransmitters released into the space that is called the synaptic cleft. The synaptic cleft is nothing more than what, people?
Free space.
What is that space?
Interstitial fluid.
Right, nothing more than interstitial fluid. And the neurotransmitter will bind to receptors on the postsynaptic cell. The postsynaptic cell, we said it's a neuron, so it's either the cell body or it's the dendrite of the neuron. That all is a synapse. So take a look once again, the two cells don't touch each other. So we would think with a synapse that like these two cells physically touch each other. They don't. And as a matter of fact, there's no proteins in the membrane touching each other as well. They just approximate one another. There's a physical space there, and they communicate with one another through neurotransmitters floating in interstitial fluid. Question? Okay. Let's look at a neuron, non-neuron synapse. So the neuron 1, we have its synaptic terminal, and now we don't have neuron 2, we have some kind of effector cell. So the best example of this is a neuromuscular junction, which we'll talk a lot about in this class. So here it is, the axon of a neuron. Here's the terminal knob of a neuron. And then this is the effector cell. That's a skeletal muscle cell. That's an actual image of one. And you can see the skeletal muscle cell has a bunch of these vertical lines here. These are called striations, and they basically are manifestations of what we call sarcomeres that are inside the muscle. We'll talk about that later. The plasma membrane of the muscle cell is called a sarcolemma. We talked about that before. And this is all sarcolemma here. This is all sarcolemma here. And so is this. But this dotted line here, I have it dotted because that sarcolemma has a special name. It's called a motor end plate. So the motor end plate is a specialized region of sarcolemma that is involved in a synapse in a neuromuscular junction. And we'll talk about the details of that later as well when we get to muscle physiology. This is actually a scanning electron micrograph of that. And you can actually see myelination on these axons here, really nice nerve terminals synapsing on skeletal muscle cells. And you can see the striations of the cells as well. Question?
So is the axon, it's still not connected to the skeletal muscle cell, right?
Well, the axon is certainly not, but yeah, I get your point. The terminal knob looks like it is, right? So if I go back here, looks like it's touching it, right? The reality is if you were standing here, you could actually swim right up to here, right out on the other side. So it doesn't look like it here, but there actually is a physiological space there.
So basically, it acts as the synaptic cleft between the sarcolemma and the axon.
Say that again.
Does it act like the synaptic cleft
Does
between
what
the,
act like the synaptic cleft?
I mean, between the sarcolemma and the axon? So
Yeah.
would it
Yeah,
that
yeah, the synaptic cleft is the space between the terminal knob and the muscle cell.
Okay.
Okay. Let's look at neuroeffector junctions. So we have neuron, non-neuron neuroeffector junctions. And so we do have a neuron, the presynaptic cell. And then again, we have an effector cell. So how is this different than this neuromuscular junction deal? Well, here, there is no really terminal knob. So we have a neuron here. There's its axon. And the axon kind of branches all over the place. And along the axon, there's these dilated regions. These dilated regions, for all intents and purposes, function like synaptic terminals, but they just don't terminate anywhere. They just kind of like daisy chain over the surface of tissue. And indeed, they have vesicles that release neurotransmitter and all that stuff. But the idea with it is to cover like a sheet of tissue so that you can activate it from multiple different regions at the same time. So you just don't have one axon with a terminal knob synapsing with one cell here. You have a bunch of neurotransmitter released here, released here, released here. We call these dilated portions of the axon varicosities. And again, they're very similar, that is, to synaptic vesicles, other than they just don't terminate anywhere. Now, all of these smooth muscle cells, they're going to communicate with each other anyways via what?
Yeah.
So go ahead. Sorry, I cut someone off.
neurotransmitters? You mean?
They're going to communicate with each other through gap junctions. So you can daisy chain these varicosities over a sheet of cells. All you have to do is activate a few cells, and they'll activate the rest of the cells by communicating through their gap junctions. So if you excite this cell, then this cell will be connected to another smooth muscle cell through gap junctions, and whatever excitation is occurring here will just travel to this cell and excite that cell. You don't need a varicosity to excite it. And by the way, that's why, for example, your heart's made up of 4 different chambers, 2 atria, 2 ventricles. That's why your right atrium, when it contracts, it does this, but it's made up of thousands of cells. So you have thousands of cells that are contracting together in unison like it's one cell. They're behaving like one cell. The reason for that is an organization like I'm describing now. That's an actual image of it, scanning electron micrograph of some smooth muscle and varicosities. Just a reminder, no images like this on the test where you have to point that out or to identify if it's TEM or SEM. Don't worry about that. Okay, we talked about excitability before, and we said that pretty much every cell of the human body is excitable because it's more negative on the inside than the outside. So here now is a number for you to memorize. Neurons have a resting membrane potential of minus 70 millivolts. What's interesting about neurons, which is not the case for all excitable cells, is neurons can be turned on or turned off. So that's really cool. And when we turn on a neuron, that means we excite it, which means we depolarize it. When we turn off a neuron, that means we inhibit it or we hyperpolarize it. So for example, you can't stimulate muscle cells to relax. You can't hyperpolarize muscle cells. The only thing you can do to your biceps is contract it. And you may say to yourself, well, no, I can lengthen my bicep. No, you can't. The only way you can make your biceps relax is contract your triceps. So we can't actively stimulate muscle to lengthen, if you will. And to do that, you'd have to inhibit it. We can't inhibit muscle, but we can turn on neurons, excite them. We can turn off neurons, de-excite them, if you will. So neurons can conduct these changes in membrane potential. notably action potentials. Well, that's not unique to them. Pretty much any excitable cell can do that. By the way, a beta cell, when glucose comes in it fires action potentials along its plasma membrane and then releases insulin. So that's not exclusive to neurons. This is pretty unique. Neurons, in response to being excited, they secrete neurotransmitters. So they manufacture, they transport, store, and secrete neurotransmitters. So that's pretty unique. And the way they transport these neurotransmitters from the cell body down to the terminal knobs is called axonal transport. So here's how that looks. We have microtubules running the entire length of the axon. And then we have these little motor proteins. So these little guys, I like to call them, they have 2 hands and 2 legs, and they walk on microtubules. And it looks like this. Now, again, we don't know, it looks like identical to this. I mean, we have a lot of evidence to support this, but you can't see something at this level, right? Because to see at this level, nothing can be alive. But we have a lot of evidence. We've tagged these proteins with fluorescent probes and stuff like that. We know they walk like this, and they walk really slow. They walk real clumsily. And you might say to yourself, well, I thought neurons respond really fast. They do. This is just happening all day long in the background in the axoplasm. These action potentials that are occurring along the membrane, that's a completely different story. What I'm talking about is making neurotransmitters here and then storing them here so that they can be released once this action potential gets to the end of the neuron. So these motor proteins, what they do is they use the energy of ATP. So they hydrolyze ATP and their feet do this. So they walk. So they carry vesicles and then walk on the microtubules. This protein called kinesin walks in that direction. We call that anterograde. Sometimes we call it the plus direction. And then dynein goes in the other direction. So it brings these vesicles back. We call that retrograde. Back means retro. And we call that the negative direction. Just as an aside, does anyone have an idea of where the plus and minus come from?
The polarity of the microtubules?
Very
Yeah.
good. So the microtubules originate in the microtubule organization center, somewhere around here, let's say, where the centrioles are. That's the negative area. And then the plus end of the microtubule is the growing end that grows towards the plasma membrane. That's why they get the plus and minus names. So this is an actual neuron image right here. If we blow up that axon, this is a transmission electron micrograph of what inside of the axon looks like. People, these are microtubules, and these are your motor proteins. So we know there are these little stick figures. We have actual images to show that. Okay, let's talk now about equilibrium potentials. So there's membrane potential at which a particular ion is at equilibrium across the plasma membrane. In other words, between the intracellular fluid and the interstitial fluid. So we're really interested in sodium and potassium in this class, so we'll talk about the equilibrium potentials for them. Let's just look at the numbers first of all. For sodium, it's plus 60 millivolts, and for potassium, it is minus 90 millivolts. And the resting membrane potential of a neuron is minus 70. So if we take a look at this, we can see that sodium wants to come into the cell. Why is there more on the outside than the inside?
Sodium potassium pump.
You got it, sodium potassium pump. So sodium wants to come in because there's more of it on the outside than the inside. We call that a chemical gradient. The sodium also wants to come in because it has an electrical gradient favoring it to come in the cell. What is that electrical gradient?
Plus charge,
Sodium's plus charge, what's the inside of the cell?
negative charge.
Negative charge. So it's got this huge electrical chemical gradient, a big vector here drawing sodium in. Potassium wants to leave because there's more on the inside than the outside. Why?
Sodium potassium pump.
Correct. But its actual electrical gradient's reversed. It's positive, the inside of the cell is negative, so it wants to stay. but its chemical gradient is so much stronger than its electrical gradient. The vector is smaller, but it's still outward. So its chemical gradient still favors the outward movement of potassium. So if we take a look over here, here we see that sodium has an equilibrium potential of plus 60 millivolts. What does that mean? Well, you can see here that we have sodium coming in through leak channels. That C stands for current. And so sodium is coming in, sodium's coming in, sodium's coming in. It's a positive charge. And once so much positive charge comes in, eventually it's going to start being pushed back out. And that point where the electrical and chemical gradients are basically equal and opposite, that's the equilibrium potential for sodium. And the same thing for potassium here. So in other words, if you were just to take a cell and let's just say an artificial cell that you created, just basically a membrane, and you put sodium channels in the membrane and you put some sodium on the inside and the outside of the cell and just said, go, do whatever you want to do. Sodium would come into the cell, come into the cell, come into the cell, and then the cell would end up at plus 60 millivolts. If you just put potassium channels in the membrane, of course, you'd have to have sodium potassium pumps as well. But if you just put potassium channels in the membrane and say, go, do what you want to do, potassium would leave, and leave until the cell reached minus 70 millivolts. So what's cool about this is this. These two numbers are very close together. So what's the number one contributor to the resting membrane potential of a neuron? What ion?
It's the potassium leak channel, right?
Yeah, even more specifically, potassium leak channels, right? So it's potassium and it's potassium leak channel. So potassium constantly leaking out of cells is what gets the membrane potential very negative. Now, the reason it doesn't get it to minus 90 is because there's many other ions that are involved in calculating the membrane potential of a neuron. You have to take into consideration sodium, potassium, chloride, calcium, magnesium, stuff like that. But it's really close. So minus 70 is very close to minus 90, and that's because largely the membrane potential is due to potassium leaving the cell. Now, the only reason potassium can leave the cell is because it was pumped into the cell to begin with. And again, you might say that's futile, but hey, check it out. It's not. It's why we have a resting membrane potential. And if we didn't have a resting membrane potential, that means two things. One, our neurons would be firing all the time, which we don't want. Two, they wouldn't be able to fire. If you can't get it from a resting membrane potential, you can't. If you don't have a resting membrane potential, then you have nothing to excite. So that's how important these potassium leak channels are.
Oh, quick question.
Yeah, go ahead.
The negative 90, is that when it hyper depolarizes? So it
Yeah, we
goes
call
more
it
negative?
we call it when it hyperpolarizes, but
Oh,
yeah,
hyperpolarizes.
but that's for an action potential. We're not looking at an action potential right now. That 90 right here is just the equilibrium potential for potassium. But yeah, in an action potential, that's a hyperpolarization. Now, if we look at resting membrane potential, there's three things responsible for establishing it. So there is number one, potassium leak channels. And you know why. Why are they the things that are most responsible for establishing the resting membrane potential? Because when they work, they get you to minus 70, which is really close to minus 90. The second thing would be the sodium potassium pump. And I don't mean because you need the sodium potassium pump to get potassium to leave, but sodium is pumping 3 positive ions out, but only 2 back in. So that leaves the cell a net what? Leaves it a net negative on the inside, right? 3 positive out, only 2 positive back in. So we sometimes say the sodium potassium pump is electrogenic. Now, in reality, it's not a huge effect on membrane potential, but for some cells it can be. But in general, it's not. But it is a contributor. The third thing would be this. The inside of the cell has got a bunch of proteins and RNA and stuff like that that are big time negatively charged. And they're responsible for what the RMP is as well. But I want you to be a little bit careful here. Let's say that a cell's at minus 70 millivolts, and let's say we open up a sodium channel and we let a bunch of sodium come into the cell, and then that minus 70, let's say it goes to minus 60. So we went from minus 70 to minus 60. Let me ask a couple questions about this. Have we depolarize the cell, yes or no?
Yes.
Yeah.
So when we go from -70 to -60, that's a depolarization, right? Now think about that. That means that -60 is a less polarized state than -70, right? Because, and the reason you know that is if you keep going this way, eventually you're going to go through a number called 0. So if you're going to go in a direction where you eventually reach 0, then there'll be 0 polarity across the plasma membrane. So that is a depolarization phenomenon here. Now, if we go from -70 to -60, I'm asking you now, list these things here, singularly or all, that can get you back now to -70. So which one of these three things, if not all, can get you back from -70 to -60 once you've been depolarized to -60?
We have to increase the K plus flow.
So can potassium leak channels get you back? I see someone nodding, Evan.
Yeah.
Absolutely. Can
Yes.
the sodium potassium pump get you back? Sure, right? It's electrogenic. It's 3 positive out, only 2 positive back in. Can these anionic molecules get you back?
Yes.
So the answer is no. Why can't they?
Or maybe they're not enough to bring it up again.
Well, the point is they don't really change. So they are what they are. So they're responsible for what your membrane potential is for whatever it is at any given moment, right? But once you deviate from that resting membrane potential, the only things that can quote unquote actively get you back would be like the movement of ions, potassium, sodium, whatever, via potassium leach channel sodium potassium pump. So these things inside the cell, like RNA and protein, they're responsible for what the ultimate value is, but they can't help you get back to resting membrane potential. Does that make sense?
Wait, could you like explain that one more time, please?
Let me ask someone else if they can. Let's do a little bit of peer education. Someone explain to us why anionic molecules can't help get you back from -60 to -70.
Could I try to do it?
Please.
So the anionic molecules stay within the cell membrane, so they are contributing a negative, like a negative charge to the overall charge, but they're not leaving and coming back, so it's not really changing.
I like that explanation. Did that help whoever asked?
Yeah, that makes perfect sense. Thank you.
Yeah, and then what is leaving and moving that will help change are these things, right? Potassium and sodium with these two different mechanisms. Thank you for that, Angelina. So look, I don't ask you guys to memorize anything really in this class, not much really, but I do want you to memorize this table. Because this is pretty important. You're going to see this a lot moving forward in your college career, in the clinic, so on and so forth. No, I'm joking. You don't have to memorize this table. I wouldn't do that to you. But
Was already writing down the note to memorize it. Oh,
cross it out and put a smiley face next to it.
okay.
So the deal is you will learn these at some point. It doesn't have to be this class. There's some things I want you to know about this, but certainly do not memorize this. So look, these are the different things that we're concerned about: sodium, potassium, calcium, chloride, and glucose, osmolar, all that stuff. We're concerned about that. These are the intracellular fluid concentrations of them, interstitial, and then plasma concentrations. Certainly don't memorize the values, but I want you to have an appreciation for like the magnitude of difference between them. In other words, we keep saying sodium's more abundant on outside of cells than inside of cells. Well, here it is. Sodium on the outside is 145 millimolar. On the inside, it's 15 millimolar. Now, just appreciate the difference in magnitude between those. You don't have to know the absolute values. We always say potassium is more abundant on the inside than the outside. Well, 120 on the inside, 4.5 on the outside. So these are the values they actually are. Look at calcium. How much more abundant is calcium on the outside than the inside? By how many orders of magnitude?
4?
Yeah, it's 10 to the minus 4 on the inside of the
Yeah.
cell, so it's 4 orders of
Or
magnitude
I
higher
guess
on the
it'd be
outside than the inside.
3 because it's 10 to the minus 4 to 1, so it'd be 1 to the minus, or maybe I have it backwards. Would it be 5?
No, it's what you said. It's 4 orders of magnitude. So that means that there's a lot more calcium on the inside than the outside of the cell, so keep that in mind. Now, with that said, with these 3, let's just do one more. Look at chloride, 116 on the outside, 20 on the inside. So it's more abundant on the outside than the inside. So have an appreciation for the magnitude differences, but really the take-home message is this. If you open a sodium channel, where's sodium going? If
Outside,
you open a sodium channel in a membrane, where's sodium going?
going outside.
It's going inside.
Oh,
It's
inside.
more on the outside than the inside. If you open up a sodium channel, a potassium channel in the membrane, where's potassium going?
Outside.
Correct. If you open up a calcium channel, where's it going?
Inside.
Inside.
You open up a chloride channel, where's it going?
Sorry.
Inside.
Correct, inside. So those are really important to know. And so these concentrations are always what they are. And that's because that our body is very, really our kidneys are exquisite at regulating these ion concentrations. Now, the last thing I want you to have one second. Last thing I want you to have an appreciation for is this. Look at your interstitial fluid. It's 290 milliosmolars, basically the concentration of solutes. Look at your plasma. It's 291. Who has the lower osmotic potential, your blood or the fluid surrounding your blood?
Surrounding?
Lower osmotic potential. Lower osmotic potential means more or less solute.
Oh, so it would be more water concentration or higher water concentration.
Higher water concentration where?
Plasma. Right? Unless I don't understand like osmolarity.
Yeah, no, the plasma has more solute, right? That's the 291 versus the 290. So where does water want to go?
To the
Plasma.
Into your plasma. So there's a constant draw of fluid into your cardiovascular system. And by the way, the main reason for that difference between the 290, 291 is a protein called albumin. So albumin is drawing fluid into your cardiovascular system so that you have good blood volume and good blood pressure. Yeah, was there a question?
Yeah, so what I'm wondering is it just says protein generally, and how is how is the concentration gradient not dependent on each individual protein? Because I mean, proteins are going to kind of have like different charges, different properties. So how can it just be protein overall?
Yeah, well, I think that's what it is. It's just the overall protein content in the cell. And we know that we just don't find proteins on the outside of the cell. So it's not a particular protein or anything they're looking at. They're looking at overall protein concentration.
Okay.
Anything else? You guys will know these numbers like the back of your hand someday. At least these ones right here. But it doesn't have to be for this class. Let's talk about changes in membrane potential. So there's our minus 70. And if we have a positive deviation from our resting membrane potential, there's a positive deviation, we call that a depolarization. I think someone just described it earlier, right? So we're less polarized. We go from minus 70 to minus 60. A return from this minus 60 to this minus 70 would be a repolarization. So we repolarize from minus 60 to minus 70. And then if we repolarize and then we overshoot that, we would hyperpolarize. So we do a negative deflection here. Now, let me ask you a question. To depolarize, give me some examples of ions that could come in the cell to cause this.
Sodium.
Sodium.
Good. What else? Sodium's the best example. What else?
Calcium.
Calcium?
Very good. What else?
Chloride.
Why not chloride?
It's negative.
Because it's negative, right? So let me
This
ask you this. What
positive
things would contribute to that?
Sorry, what'd you say?
What things would contribute to that negative deflection?
Hyperpolarization, you'd use chloride?
Yeah, what ions?
Chloride.
Why?
Chloride. And then what else?
Potassium
Think
leak channels.
about this a little bit. Go ahead.
I said potassium leak channels.
Yeah, potassium leaving the cell, right? So
Yeah.
potassium leaving the cell can hyperpolarize the cell as well. Good. Let's look at graded potentials and then we'll look at action potentials. So a graded potential is a change in membrane potential. The change is the delta sign, and then don't worry about it. You won't have to do any calculations, but V sub M means membrane potential. So a change in membrane potential, or the magnitude of it, or the amplitude of
You
it.
answer the phone the same way every time. Can you please go save my number?
Oh my. So where the magnitude or amplitude of it is variable, but directly proportional to the size of the stimulus. So it's kind of a mouthful, but let's take a look at that. So this is just some random strength of stimulus here. So we have some kind of stimulus. And what that means is we're releasing a certain amount of neurotransmitter, let's say. And that neurotransmitter is going to open up a certain amount of channels. And when it opens up a certain amount of channels, a certain amount of ions are going to come in the cell, and then we're going to get that. We're going to get a depolarization. But if we have a lot more neurotransmitter released because we're firing that neuron with more frequency, then much more channels are going to be activated and much more current's going to come into the cell. We get that, and then so on and so forth. So in other words, the strength of the stimulus is directly proportional to the magnitude of the change in membrane potential. It's very different than action potentials. Action potentials will always go from -70 to around +30.
I have a
Oh,
quick question.
One sec. So the amplitude's always fixed, but in a graded potential, the amplitude depends on the amount of current that's coming into the cell. Yeah, go ahead.
Just when you depolarize, the sodium goes out or in?
In the cell.
Okay.
Remember, there's always more sodium on the outside than the inside. Questions on this?
Professor, you said the amplitude is fixed when it's not a graded potentials?
Yeah, in action potentials. We'll
Oh,
talk about
action
them in a
potential.
minute.
Okay.
Yeah, action potentials, they're fixed. They're variable in graded potentials.
Okay, thank you.
Indeed. So how do we generate these graded potentials? Very important for you to remember here. By the opening of mechanically or ligand-gated channels. Note what's not on here, voltage-gated channels. If you open a voltage-gated channel, that's going to give you an action potential. If you open up mechanically or ligand-gated potential channels, that is, you get graded potentials, not action potentials. So look, here's a postsynaptic neuron. And if we take a look at its membrane there, we can see that there's a protein in the membrane. That's a ligand-gated sodium channel right there. And you can see that acetylcholine binds to it. Actually takes 2 acetylcholine molecules to change its conformation to an open state. And it's called a ligand-gated sodium channel because when the ligand binds, which is acetylcholine, the receptor becomes a channel permissive to sodium. So think about how cool this is. Okay, acetylcholine's been released because you have a presynaptic neuron that is releasing it from its synaptic vesicles. It binds, changes the conformation of the protein, and it opens. And sodium comes in because it's permissive to sodium, but it doesn't come in, sodium that is, just because the channel is permissive to it. It comes in because there's a gradient. So what I'm trying to say to you people is this: the sodium potassium pump is money, because if it didn't run all day long in the background, you could fire your neurons all you want, and you can release all the acetylcholine you want, and you can open up all the sodium channels in your muscle, but they would never contract because sodium would never come in. You need sodium to come in a muscle cell for it to ultimately contract. And sodium always will come in the muscle cell if you open the channel, because there will always be more sodium on the outside than the inside. Look, there are toxins that inhibit sodium potassium pumps. And if you give enough of that toxin to someone, you will kill them because you won't be able to fire action potentials. So you won't be able to live. Believe it or not, the drug is given to some individuals for certain reasons, but obviously in very low concentrations. So this is a mechanically gated channel. It's closed. You push your finger on the membrane, it opens. So a good example of this would be like in the cervix with those mechanoreceptors. And again, ion will come in and you'll form action potentials. Questions on this? So graded potentials are sometimes called local potentials. And the reason we sometimes call them local potentials, remember graded is because the amplitude of the change in membrane potential is variable. Why we sometimes call them local potentials is because basically what a graded potential is this. You open a sodium channel, sodium comes in, there's a huge concentration when it first comes in, but then the gradient starts to get smaller and smaller as it starts to diffuse down its concentration gradient. Now, that's a gross exaggeration in this image. The reality is it would look something like this. A bunch of sodium would come in here, and then it would move this way, this way, this way, and that's about it. That's about the amount of membrane it would affect. So in other words, it's a very local change in membrane potential. It's not going to spread very far. So in other words, if you open a channel here and some sodium come in, you'll get a little bit of change in membrane potential, maybe here and maybe here. That's it. So it kind of looks like this. The point of origin where you, so to speak, inject current, which is sodium, into the cell, you can see that there's a huge change in membrane potential. This is amplitude of membrane potential here, huge. But if you look outside of that, it starts to exponentially decrease over distance. So as soon as you start moving this way or start moving this way, that change in membrane potential precipitously drops because diffusion is distance limited. So it's kind of like throwing a pebble in a pond. The bigger ripples, then the smaller, the smaller, the smaller. So in other words, graded potentials decrement. They get weaker over distance. And that's why all blood vessels are very close proximity to cells, right? How close are they to cells?
20 microns.
Yeah, about 20 microns. If you get them any further than that, then the diffusion of nutrients or waste away from the cells is not efficient because diffusion is distance limited and the cells die. Questions on this?
Professor, so what's the difference between action potential and the graded potential mainly?
We're getting there. I'm being a little suspenseful. Not really though, because I need to describe graded potentials before we get to action potentials because there's a very important link to the two. And let me say it now, and maybe you can write it down. Graded potentials initiate action potentials. Graded potentials initiate action potentials. So that's why I need to talk about them first. All right, here's action potential.
Oh, sorry.
No problem.
Good question. You said graded potentials decrement, they decrease over time or they go down. So like they like the ripple, you said it increases to a high point and then it just goes straight down. Did I hear that? Okay, I just wanted to make sure I
Yeah.
heard that.
And what that means is that the concentration gradient's getting smaller, smaller, and smaller. Okay, action potential is this rapid localized change in membrane potential. They're variable in frequency, but fixed in magnitude. They result as the opening of voltage-gated channels, notably voltage-gated. Sodium channel we'll largely talk about. So there is the action potential now. And there's our minus 70 millivolts resting membrane potential. And you can see this is a new term for you. We have a threshold potential of minus 60. What does that mean? That's the potential at which voltage-gated sodium channels open, and they open right here. Now, we're at minus 70, and we want voltage-gated sodium channels to open here. How are we going to get from here to here? Something's
Graded
causing us to
potential?
go from minus 70 to minus 60, so it is a depolarization. What's causing that?
Is that the graded potential?
You got it. Graded potentials are causing that little drift. And then that little drift gets you to -60, and now voltage gated sodium channels open. And now there's this huge influx of sodium into the cell, and that's this whole upward leg right here. But what happens is it stops at +30. Now it's not the same in all neurons, but generally speaking, that's fine. But it's not variable. You can't change that. It's an all or none deal. When those channels open, it's going to go to +30 and that's it. Now, the reason it's going to stop here is because at +30, sodium channels inactivate. And we'll get to that in a little bit. But what also happens at the same time the sodium channels inactivate, voltage gated potassium channels open. And now what happens is potassium leaves. And now you rapidly repolarize. And as someone mentioned earlier, what happens with these potassium channels is they stay open a little bit longer. I often say they stay open longer than they should, but it's not like they're violating any law. They're just slow to close, we say. And so the cell actually goes down to -90, and then it drifts back to -70. Probably sodium leak channels get it back to -70. So we call that a hyperpolarization right there. Some people really like to talk about this after hyperpolarization. It's important, but I mean, people get a little bit too interested in it, too excited about it. Now, please appreciate that the length of this action potential was about 3 milliseconds. So think about how rapid that is. It's a very fast phenomenon. Also appreciate that that means that that's the duration of an action potential. But I mentioned a term up here called frequency. And what that means, if this action potential is occurring right here in this one node of RANVA, if you had an instrument here to measure how many action potentials show up at that point along the axon per unit time, that would be frequency of action potentials. So how many action potentials occur at a given node of RANVA per unit time, that would be frequency. But we're not looking at that right now. We're looking at the length of an action potential. That's duration. And by the way, what's this?
Amplitude.
You got it. So now you got three components right there. You got the amplitude, you got frequency, you got duration. Now, let me tell you something about this frequency deal. This frequency here is the code of basically the neuron. And what I mean by that is if you have a neuron and it goes blip, 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 versus if it goes blip, 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 blip. So if it goes faster, in other words, you have more action potentials occurring per given time at a node of RANVA. That's the difference between looking at my screen right now and seeing it white, very bright, and maybe me dimming it and you say, well, that looks a lot dimmer now. That's the difference in code. How can you discriminate between the difference in intensity of light? Well, higher intensity would be greater action potential frequencies. Lower intensity would be slower action potential or lower action potential frequencies. And we'll talk more about that later.
Could you explain, you were saying like, you said it really quickly, so I didn't get it really quick, but you were saying like measurements of different frequencies at different speeds per something?
Yeah, if you're able to measure in a node of RANVA, which we can measure how many action potentials occur per unit time in a node of RANVA. Every time you would see a blip there.
Did you say RAN VA? Did I hear that correctly? RAN VA?
Yeah, node of RANVA, yeah.
Okay.
Yeah, so every time we would see one show up there per unit time, that rate would be frequency. I have a slide to illustrate this. I'll show it to you a little bit later.
Okay, thank you.
Indeed.
When you repolarize, potassium leaves, right? It goes outside the cell.
Correct.
Okay.
So are the voltage gated sodium channels like located in the node?
That's a beautiful question. And they are. So the node of RANVA is loaded with voltage gated sodium channels, voltage gated potassium channels, and what else has to be there? So let me say that again. A node of RANVA has to have voltage-gated sodium channels, voltage-gated potassium channels, and what else has to be there?
Sodium, potassium.
Potassium leach
Beautiful.
channel?
Sodium potassium pump. How about mechanically gated channels? Or ligand gated channels? Would they be in a node of RANVA?
No.
Absolutely not, because we don't have graded potentials occurring there. We have action potentials occurring there.
The length of graded potentials.
Sorry, go
So
ahead.
the length of graded potentials is shorter than the length of or duration of an action potential, correct?
And not the duration, I get what you're saying, but it's not the duration per se. Like you wouldn't look at a graded potential, like here's the action potential duration. You wouldn't look at a graded potential and say its duration is like shorter that way.
Like the time that it takes though, like if you're looking at
Yeah,
the
yeah,
bottom.
I get your point. And yeah,
Right.
what you're thinking is correct.
Okay.
Yeah,
Thank you.
indeed. All right, let's talk about this concept called refractoriness. So basically what it is, the unresponsiveness of a neuron to stimulation while it's already being stimulated. So there's two different components of the refractory period of a neuron. There's the absolute refractory period and then the relative one. So we'll come back to this thing in a second. So this is the absolute refractory period here. So what does that mean? Well, if a neuron's undergoing an action potential here, you can't undergo another action potential while you're undergoing an action potential. So that means the neuron's completely refractory. You could stimulate the neuron with neurotransmitters, but it's not going to do anything because it's already undergoing an action potential. So in other words, voltage-gated sodium channels are open. You can't open them, or you can't open them more. Now, the relative refractory period is here, and I'd like to bring it over a little bit. So we'll just draw a line here, a little bit over here as well. And what that means is that in this region right here, you could potentially undergo another action potential. The reason for that is as you're repolarizing, voltage-gated sodium channels are starting to reset. They're starting to close. And they start to close somewhere down here, so you could potentially re-stimulate here, and certainly where you're hyperpolarized, they're closed, and you could undergo another action potential here. So that relative refractory period, though, it would require a really strong stimulus to overcome -90 to get to -60, as opposed to -70 to -60. So you would need a stronger stimulus, but theoretically you could do it. By having refractoriness, this sets up two things that are interesting. One, it determines the direction of action potentials. So if an action potential is traveling like this, that means that if it's refractory here, then the action potential can only go in one direction. So in other words, as you have an action potential that occurs, let's say in a node of RANVA, let's draw a couple of them. So you have an action potential that occurs here. The neuron's going to be refractory here. And so as a result of that, the next action potential will occur here, not here. So in other words, sodium comes in, potassium leaves, you're refractory here. That means current will flow over here, open your sodium channels here, you get another action potential here. So action potentials go in that direction. They don't go in the opposite direction back to the cell body. The other thing that it does is it sets up the maximum frequency that action potentials can occur. So let me show you this. Well, theoretically, we could stimulate another action potential here, but let's just say we wait to here. So here's our next action potential, and then our next one occurs, and then our next one occurs. That's action potential frequency, people. That's the duration of one, that's the duration of another, that's the duration of another, but that's the frequency of action potentials. But here's my point. Because of the refractory period, we can't get the action potential to get this close together. That's a really high frequency. We can't get that frequency because of the refractory period. We can't fire another one till here. So that sets the limitation of the fastest frequency that we can have, or the highest frequency that we can have. Questions on that?
Do you know if there's any variability like between like either individual like humans or like, you know, certain animals and stuff? Like I'm just curious like if that's like a pretty set frequency or if there's like variability like even within different like
Oh yeah, definitely
neurons
variability.
in the body.
Yeah, and there's variability within our body that we'll talk about. That's really cool.
Yeah, that was the last part.
Yeah. And by
Interesting.
well, there's
Okay.
there's another thing that I'll tell you in an aside. But yeah, there's variable within the body too. We'll look at that in a second. Actually, we're going to look at it right now. So here's action potential frequency. And if we look at the a neuron in the brain, it's about 300 hertz. We look at a skeletal muscle, it's about 200 hertz. And look at cardiac muscle, it's about 5 hertz. So really slow in cardiac muscle. We'll have to take a look at why. So if we look at 1 cycle per millisecond, and we say there's 1,000 milliseconds per second, and then 1,000 cycles per second, that's 1,000 hertz. And if we get rid of the term cycle and we make it action potential, then 1 action potential per 2 milliseconds is 500 hertz. Well, we said it's about 3 milliseconds. So what's 1 action potential per 3 milliseconds? How many hertz?
333?
Yeah, it's like 333 or something. So it's this, right? Brain neuron. Now, feast your eyes on this. This is action potential duration in a brain neuron. Here's action potential duration in a skeletal muscle cell. And look at action potential duration in a cardiac muscle cell. So the cardiac muscle cell, the action potential duration is 200 milliseconds, not 3 milliseconds, 200 milliseconds. Now, in a brain neuron, because the duration, let's say, is 2 to 3 milliseconds, you can get another action potential here and another one here and another one here, really close. And for a muscle, they're a little bit further spread out. But look for cardiac muscle, your next action potential is going to occur way over here. So that's why the frequency is really slow, 5 hertz versus 333, right? So the hertz is really slow because the duration is so long. But here's the deal. We know that when we stimulate a neuron to stimulate muscle to contract, that when we get an action potential in muscle, we'll get a little bit of tension build up in the muscle. But if we fire another action potential repeatedly, and then another one, and then another one, then we get these series of action potentials that cause a sustainment in that depolarization and that tension. So much so that we get a tetanus. So in other words, if you can bring these action potentials really close by firing that neuron repeatedly, then in a muscle, you can get sustained tension, which is what we want. But we don't want that in our heart, right? We don't want our heart to squeeze and then hold like a fist for any period of time. And it could if it had a shorter duration. So if it had a shorter duration and we could fire another action potential, we could die because our heart would stay contracted for a sustained period of time. But it can't. It's impossible for it to because its refractory period is so long that it can't fire another action potential to here. Now, what's not shown here is during this action potential, which is an electrical event, the muscle is also going to be generating tension. And that tension curve looks like this. And what that means is the amount of tension in the muscle, the amount of force in the muscle, by the time you can stimulate it to contract again, is 0. So by the time you can stimulate a muscle cell to contract again, it doesn't have any tension in it. So it can't summate. So you can't have a tetanus in the heart.
Wait,
Thanks.
sorry.
Yeah, could you
What?
repeat that again?
Heart doesn't hyperpolarize? Is that what you're saying?
Say that again.
Are you saying the heart doesn't hyperpolarize?
Um,
You said it goes to zero. Confused about that.
well, one sec. Okay. No, this has nothing to do with hyperpolarization. What I'm saying is that the heart has a really long duration, and during that duration of the action potential, muscle tension rises and then muscle tension goes to zero. So that by the time you can stimulate the muscle to contract again and generate another action potential, there is no preceding tension. So for muscle to generate a sustained contraction, like when you exercise, you have to have a little bit of force and add more force to it and add more force to it. You need this summation to generate a tetanus. But the heart can't do that because we can't get the action potentials close enough together like we can in muscle.
So does that place, like, you would be in a bad way long before this, but does that place kind of like a theoretical upper limit of 300 beats per minute for how fast your heart can beat? Because
Yes, it actually
5
does.
per second would be 300 a minute, right?
A very nice calculation and very nice thought. Yes.
Okay.
We put it at 250, but yeah, it's around
A
300.
theoretical upper limit, yeah.
Yeah. Yep.
And there's
And as a
other,
matter of fact, the only reason it's not 300, and we'll talk about this later, is because the AV node in the heart doesn't allow the signal to propagate that quick through there. So it
yeah,
actually
I was thinking there's probably
tethers it
other
a little
limiting
bit.
factors too.
Yeah, because I was gonna say is the reason it has like a long refractory period. That's the reason why it doesn't go very fast. Like, is that part of because it's just a heart muscle, or do most muscles have a feature like that, but they can extend past their limit? Like, how
No,
does
the importance of it having a long refractory period is so that it can't form tetanus.
right? But other muscles can do that because they can tolerate it, and they like if
Well,
I'm lifting weights, it's okay. But the heart has a specific amount of time, like it can't
correct.
beat,
Correct.
right?
Well, yeah, what
So
we're going to learn later is that when we think of the heart, we always think about it contracting, but actually the heart spends more time relaxing than it does contracting. So yeah, it's very important that the heart can relax and fill with blood. It can't have this sustained contraction like skeletal muscle.
What about smooth muscle?
thank you.
Don't know much about it.
Okay, now I'm extra curious.
All
I'll
right.
have to look into